So we are now going to discuss about the DNA repair. Now, what is DNA repair? As given above, DNA repair is a collection of processes by which a cell identifies and corrects the damage to the DNA molecules that encodes its genome. So DNA repair is a collection of processes accomplished by various proteins, enzymes, by which a cell will identify the damage, where the damage occurs within the DNA, and then rectifies and corrects the damage to the DNA molecules that encodes its genome. Now, in human cells, both normal metabolic activities and environmental factors like radiation can cause DNA damage. Both normal metabolic activities and environmental factors like radiation can cause DNA damage, resulting in as many as 1 million individual molecular lesions. What are lesions actually? They are molecular lesions are actually damage to the structure of a biological molecule. Like here we are talking about DNA. So damage to the structure of DNA can be called as lesions. So due to some radiations, also called as physical mutagens, radiations like ultraviolet rays, especially the UVB rays, which have a specific wavelength that has the ability to cause damage to the DNA, can cause change in the structure of the DNA, and other rays like your X-rays. Then some chemical mutagens are there, like your some alkylating agents, some intercalating agents that can cause DNA damage and result in molecular lesions, means damage to the DNA. About 1 million molecular lesions can occur in the cells per day. Many of these lesions have caused a structural damage to the DNA molecule and can alter or eliminate the cell's ability to transcribe a gene that the affected DNA encodes. Actually, it causes the structural the lesions actually, which are caused by the physical mutagens or chemical mutagens like your alkylating agents, X-rays, UV rays. They cause structural damage to the DNA, and thus they can alter or eliminate the cell's ability to transcribe that gene that the affected DNA encodes. If that particular segment of DNA getting damaged and get altered, that particular segment will be further transcribed to form a different, maybe a different protein, maybe a different amino acids. Sometimes it fully eliminate that particular cell. Other lesion includes your potentially harmful mutations in the cell's genome, which will affect the survival of its daughter cell after it undergoes mitosis. There are some lesions that can be very harmful, mutations in the cell's genome, which will affect the survival of its daughter cell even if it undergoes mitosis. As a consequence, DNA repair process is very much required. It has to be constantly active. So only this mechanism is active. Neither your replication, nor your transcription or translation, nor your homologous recombination. The DNA repair process is constantly active, is constantly rectifying the mistakes that may occur during replication process. They have proofreading activity, but sometimes they misincorporate wrong bases. They may also repair the mistake. They may also repair the damage bases which has been done as a result of physical mutagens, as a result of chemical mutagens. Okay. Now when this normal repair process fail, suppose DNA repair process cannot able to do this stuff, cannot able to rectify, cannot able to correct that mistake, then what happen? The cell will undergo programmed cell death, which is known as apoptosis, right? Okay. So cellular apoptosis will occur only when your normal repair process fail. Okay. Next, it is saying that when normal repair process fail and when cellular apoptosis does not occur, irreparable DNA damage may occur, including your double-stranded breaks and DNA cross linkages, which can be very hazardous. Suppose cellular apoptosis does not occur, the repair process also have failed to correct that mistake. So as a result, double stranded, including your double stranded DNA breaks, it is very harmful. Your both strand has been cut off, has been nicked off by some mutagens. Then DNA cross linkage means when your some agents, some chemical agents, it may be exogenous agent or endogenous agent, will react with your two nucleotides within the DNA, either in the same strand or in the opposite strand, and forms a covalent linkage between that nucleotides. So again I repeat, cross-linking of DNA means when various exogenous mutagen or endogenous mutagens, they are causing the DNA where your two nucleotides may be on the same strand of DNA or in the opposite strand of DNA, because DNA is double stranded. So two nucleotides, they cross link with each other to form a covalent linkage between them, which cannot be broken down. This cross link can occur within the same strand or in the opposite strand. Okay, here it is saying inter-strand cross-link. It is saying inter-strand, that means within the same strand, that means within the two strand. Inter-strand means between the two strands, as DNA is double stranded. So due to this cross-link, it will cause various damage to the DNA, and in future it may lead to malignant tumors, it may lead to cancer. Okay, so that is why this your DNA repair is very much important. If DNA repair fails to correct that mistakes, to correct that damage base, then this type of DNA cross linkages, or it leads to tumor, it leads to cancer, right, which are very harmful to the cell and also to the body of that particular organism. Now the rate of DNA repair is dependent on many factors. DNA repair is dependent on many factors, including the cell type, the age of the cell, and the extracellular environment where it is occurring. How much the DNA will repaired, that is dependent on many factors like your cell type. The age of the cell also varies. With increasing age, your repair mechanisms will be lacking, and that leads to aging, including your cell type also, and on which environment it is facing. A cell that has accumulated large amount of DNA damage, or one that is no longer effectively repair damage, incurred to its DNA can enter in one of the three possible states. Suppose a cell which has a large amount of DNA damage, no repair system, no repair mechanism can correct that damaged DNA. Then what will happen? The fate of that cell may be one of these three. One is an irreversible state of dormancy known as senescence. Senescence is also called as biological aging. It is the gradual deterioration, means slowly it is getting degraded of your functional characteristics. So senescence, so one of the way is, suppose a cell cannot able to correct that mistake, cannot able to repair that damaged DNA, so much damage occurs within the cell. Then what will happen? The first possible that may occur with of the cell's fate is irreversible state of dormancy. The cell may get dormant, inactive, known as senescence. Second, it may undergo apoptosis, also called cell suicide or programmed cell death. Obviously, apoptosis is a very important pathway for the cell, right? Apoptosis is a very important pathway for the cell. It occurs in a series of biochemical events, right? And third thing that may occur within the cell is formation of tumor, formation of malignant tumor which may be cancerous. It may lead to unregulated cell division if it not checks, which can lead to formation of tumor that is cancerous. So these are the three fates of the cell. These are the three fates of the cell, that if the cell containing many damage, okay, cell containing large amount of DNA damage, or one that is no longer can be repaired, then these are the three possible ways that cell may enter. First, it may enter to senescence, making dormant state irreversible, which cannot be, that the cell cannot be again come back to its normal form. It permanently gets into G0 state, okay, the G0 stage of cell cycle, permanently inactive, senescence. Second, cell suicide, also known as apoptosis or programmed cell death, or it may become tumor, that the tumor become malignant also. Unregulated cell division may occur, which leads to the formation of tumor, and if the tumor is malignant, it leads to cancer. Okay, so these are the fates. That's why it's very important for us, for the cell to repair the damage, because further the cell will be divided, and if there is damage to the DNA, and that DNA damage due to which it mispairs with some other bases, wrong bases, then what happened? The cells, the gene's identity has been changed actually, right? So DNA repair ability of a cell is very vital to the integrity of its genome, and thus to the normal functionality of that organism. DNA repair ability of a cell is very vital to the integrity of its genome, and thus to the normal functionality of to that organism. Many genes that were initially shown to influence lifespan have turned out to be involved in DNA damage, repair, and protection. Many genes which have initially shown to influence the lifespan, those genes which are related with the lifespan of that organism, they are involved in DNA repair, okay, and protection. Now there are many ways through which you can repair your DNA damage, and one of the way is your direct reversal. The first way is your direct reversal. Directly it will mend the damage, directly it will move out the damage base, and or directly it will repair that damage. It will not move out, direct it will correct that mistake. So cells are known to eliminate three types of damage to the DNA by chemically reversing it. Now, cells can cells can eliminate three type of damage to their DNA by chemically just reversing that reaction. There is some type of damage to the DNA which can be cured, cured by reversing that reaction. That way is your direct reversal. There is no such thing that you have to cut off that damaged base, replace with a new base and fill with a new base, right? Not like that. It will directly mend that damage by just reversing that reaction, which leads to this mutation. These mechanisms do not require template. Now direct reversal, they don't need any template. They don't need any template that yes, we have to for this nucleotide we have to attach this nucleotide, okay, which is complementary. There is no such thing, because directly it is reversing that reaction that leads him to cause that mutation. It will just reverse that reaction to become normal base. So these mechanisms do not require any template because the type of damage they counteract can occur in one of the four ways. Such type of reversal mechanisms are specific to the type of damage incurred and do not involve breakage of the phosphodiester backbone. So this type of direct reversal mechanism, just remember, these direct reversal mechanism do not involve breakage of the phosphodiester bond. They do not break the phosphodiester bond, neither they remove out the damage base. It only mends and reverse that reaction so that damage base become again normal base. Best example, like your pyrimidine dimers, it may occur due to the ultraviolet light. Due to the action of ultraviolet light, it may lead to pyrimidine dimer formation. Now, what are pyrimidine dimers? You know about pyrimidine. Thymine or cytosine are the pyrimidines. Pyrimidine dimer means either thymine or thymine cytosine may join together by a very strong bond that leads to dimer formation, and this occur in the same strand. Suppose within the same strand there is thymine and next to the thymine there is another thymine. So due to the action of UV light, it catalyzes to form a bond between these thymine nucleotides and form thymine dimers or thymine cytosine adduct, thymine cytosine dimers. Okay, so it results in an abnormal covalent bond, which is a strong bond between the adjacent pyrimidine bases. Okay, for example, I am trying to show you here. Suppose there is thymine here, there is another thymine here. So due to the action of your UV light, it leads to the formation of a covalent bond between them, and this is known as thymine dimers. Okay, so mainly UVB light is responsible for this UVB light. Okay, so coming to the main portion now. So these pyrimidine dimers are often formed by UV light that result in an abnormal covalent bond between them, between the adjacent pyrimidine base of the same strand. Now there is a repair mechanism. I have said about this direct reversal. So one of the mechanism can be, it can be cured. It can be again the thymine dimer which has formed, it can be again reversed back to form a normal thymine dimer, a normal thymine residue, okay, by the action of photoreactivation. Okay, so the photoreactivation process directly reverses this damage by the action of enzyme called DNA photolyase. Here the enzyme has been says only photolyase. Okay, its name is DNA photolyase. What does DNA photolyase do? DNA photolyase is a repair enzyme that can able to repair the damage caused by UV light. These enzymes, DNA photolyase, they need they need some energy. They need some energy absorbed from the blue light or UV light. See, the same UV light it is using 300 to 500 nanometer wavelength to promote catalysis to break that thymine, to break that covalent bond between that pyrimidine dimers, right? So I can show you the figure. Let me pass this thing. Okay. Yeah, this is your photo reactivation. See, first picture is what you see here. Normal DNA, due to the action of UV light, it forms a pyrimidine dimer by the action of UV light. The main structure has not been shown. I will show you in the next figure. Then come DNA photolyase. They are inactive in the dark, but for its activeness they needs visible light. Visible light means 300 to 500 wavelength, right? And with the help of visible light, DNA photolysis can break that covalent bond between the thymine here is shown, and it reverses that reaction, right? So this is the photoreactivation repair process, right? So photoreactivation, what it is saying here, ultraviolet, let me show you. Ultraviolet irradiation causes the formation of thymine dimers. Irradiation can cause formation of thymine dimers. Upon exposure to light, DNA photolysase break that ring formed between the dimers to restore the 2-thymine residue. So it is just reversing the reaction by using the visible light. These DNA photolyase, or simply called the enzyme photolyase, can able to reverse that reaction so that this pyrimidine dimer again become normal. Okay, so this was about the photoreactivation repair. Okay, let me show you how the thymine dimer occurs. Okay, so this is a thymine. You can see here it shows the structure of thymine here. Okay, now you have to show the thymine dimer. So UVB light is acting to form thymine dimer. See, two covalent bonds have been formed. Okay, and thus this thymine dimer formed due to the action of UV light. Okay, so we are talking about direct reversal process. So the photoreactivation done by DNA photolysase, whose activation is dependent on your energy absorbed from blue light or UV light. 300 to 500 nanometer wavelength of light is required by this photolysase to promote catalysis. Photolysase is an old enzyme present in bacteria, fungi, in most animals, but it is no longer function in human don't have this photolysase to able to do the function. Human have a different type of enzyme to repair this type of pyrimidine dimers. Okay, human use actually nucleotide excision repair mechanism to repair the damage from UV irradiation. Humans normally uses nucleotide excision repair mechanism. As the name suggests, nucleotide means the nucleotide which is damaged. Excision means to cut off, to remove that nucleotide. Repair, in that place it will enters a correct nucleotide respect to that template strand, right? Okay, so humans don't have this type of photoreactivation repair. Okay, they don't have this type of photoreactivation repair. Another direct reversal repair is your methylation of guanine. Let me show you. So another type of damage, another type of damage is your methylation of guanine. Methylation of guanine bases. Methylation of guanine bases may occur, and it can be directly reversed into a normal guanine by the protein called methyl guanine methyl transferase. So it is saying that, what it is saying, another type of damage, methylation of guanine bases. This is a type of damage, methylation of guanine bases. It can be directly reversed to form normal guanine by the action of a methyl transferase enzyme known as methyl guanine methyl transferase. In short, it is called MGMT, where the bacterial equivalent of which is called OGT. Now it is means it is similar with OGT. Okay, it is similar with OGT. Actually, OGT is if I say what is OGT, let me talk to you. It's a bit of lengthy. Okay, so OGT, the full form of OGT is O6 alkyl guanine O6 alkyl guanine DNA alkyl transferase. So much big name. 2. So this is the full name of your OGT, O6 alkyl guanine DNA alkyl transferase 2, and it is saying here this MGMT, methyl guanine methyl transferase, is equivalent to the bacterial enzyme called OGT, O6 alkyl guanine DNA alkyl transferase, the same enzyme. This enzyme, this OGT that we are talking here, it is that enzyme methyl guanine methyl transferase MGMT is showing that its function is similar with OGT. Normally this OGT is responsible for removal of the alkyl groups. Like here this MGMT is removing the methyl group, the OGT will remove the other alkyl groups, right? Like it will remove the alkyl group from O6 alkyl guanine as done by MGMT. It only removes the methyl, it will remove the ethyl, propyl, okay, such a type of alkyl groups. It will also remove the O6 alkyl thymine. Okay, But here is saying, okay, so this is actually the main function to remove the alkyl groups and make them a normal base, but it is very expensive process. Why it is very expensive process? Because each time this enzyme will be used only once, it will transfer that methyl group into its own catalytic site, and it will be not recycled again. So we are sacrificing these mgmt molecules for correcting the mistakes. I will show to you here. Let me show you the figure. Yeah, this is the figure. So this is your methyl transferase mgmt. You can see the guanine has been attached with a methyl group O6 methyl guanine, and a enzyme methyl transferase MGMT, methyl guanine methyl transferase. You can completely write the name here also. You can also write only methyl transferase, that's no problem. So this methyl transferase having this SH functional group of cysteine, they are removing this methyl group from the guanine and introduce that methyl group into its own catalytic site, which cannot be used again, but it corrects that mistake. So it is saying here methyl transferase, or full name is methyl guanine methyl transferase, because here it is removing the methyl group from the 6th position of guanine. That's why it's an O6. So methyl transferase is catalyzing the transfer of the methyl group from the on the O6 methyl guanine to a cysteine residues on their enzyme, on this enzyme, and thereby restoring the normal guanine in the DNA. It is not cleaving or removing the damaged base, it is just reversing the reaction. This is called direct reversal. Okay, so here you can see this methyl transferase is removing the methyl group, the mgmt, okay, from the guanine. It is removing the methyl group, and it is removing, and that methyl group is attaching to its cysteine residue of that enzyme, and thereby it cannot be used again, but it is restoring the normal guanine in the DNA. So this is the thing. Okay, so this was about the um, thing about the direct reversal. The direct reversal can be done by various ways, by the help of the photoreactivation repair system, then your then by the methyl transferase enzyme, which looks like OGT, okay, in case of bacteria. Now we will go for single strand damage. Single strand damage means suppose there is damage to only one strand, not in both the strands. Then also there is various repair mechanism. Let us see. When only one of the when there only one of the two strands of a double helix has a defect, then the other strand can be used as a template to repair, to guide the correction of the damaged strand. Again I repeat, when only one of the two strands of a double helix have a defect, then the other strand can be used as a template to guide the correction of the damaged strand. In order to repair the damage to one of the two paired molecules of DNA, there exists a number of excision repair mechanisms that will remove the damaged nucleotides and replace it with an undamaged nucleotide complementary to that found in the undamaged DNA strand. Simple thing. Here you are looking that is very complicated. No, simple thing. Suppose there is damage to the DNA in one of the two DNA strands, then what they will do, they will remove that damaged segment from that DNA strand, and by by using the undamaged DNA strand as a template, it will help them to guide the correction of that damaged strand. It will place the right nucleotides complementary to the template DNA which is undamaged. This is called excision repair. Excision means to cleave off the damaged base and replace it with correct base by using the non damaged strand as a template. Okay, there are various mechanisms for this, like your base excision repair, in short it is called BER. Then your another one is there, which is your nucleotide excision repair, or NER. Then okay, this is very important, mismatch repair. So these three repair systems are actually involved as single stranded breaks. I will just talk you up to this much, up to the mismatch repair. Okay, in the next video I will discuss about the other mechanisms of the DNA repair. So let me talk about one by one. So base excision repair, the damaged single bases or nucleotides are most commonly repaired by removing the base of the nucleotide involved and then inserting the correct base on nucleotide. Simple thing, damaged base or nucleotide, most commonly they will be repaired by removing the base or the nucleotide involved. We have to remove the nucleotide damage base and then insert the correct base or insert the correct nucleotide. This base excision repair works in a step by step manner, and that is done by this enzyme DNA glycosylase. DNA glycosylase enzyme, they first remove the damaged base from the DNA by breaking the glycosidic bond from the sugar, from that deoxyribose sugar. So DNA glycosylase, I will show you the picture just now, but let me talk to you about this repair mechanism, the main principle. DNA glycosylase, they first, as the name suggests, glycosylase, it's easy to remember. Glycosylase means, lase means enzyme, glycosylase means glycosidic bond. So it breaks the glycosidic bond, of the damage base and remove that damage base, right? Okay, and remove the damage base from the DNA by cleaving the bond between the base and deoxyribose sugar. Then the other enzymes will remove, so it becomes a episite. Okay, episite means either it may be apurinic or apyridiminic. Okay, it may be apurinic or apyrimidinic. Apurinic means if it was a purine that has been removed by the DNA glycosylase, it will be, because there is no purine there, only the sugar and the phosphate group is attaching, the base has been removed by the glycosylase enzyme. Okay, it becomes apurinic or apyridiminic, means if there was a pyrimidine which has been damaged and has been removed by this glycosylase enzyme, it is baseless. It has no base, so it is apyridiminic. Both are baseless sugars. Okay, and then further there are some enzymes called epi-endonuclease which will remove the rest of the backbone of the site. Then some sort of repair DNA polymerase will remove the damaged region using its exonuclease activity and correctly synthesize the new strand using the complementary strand as a template, and the gap will be finally sealed by the enzyme ligase. So first, DNA glycosylase, this enzyme remove the damaged nitrogenous base while they will leave the sugar phosphate backbone intact. Thus it creates a episite, apurinic or apyridiminic site. Okay, next this episite will be further removed out, can be removed out by the epi-endonuclease which will remove the rest of the nucleotide, and DNA polymerase then remove the rest of that. It trim that damaged region. If there is any damaged region, it trim that region, DNA polymerase, by its exonuclease activity, and finally it will correctly synthesize that also in the new strand using the complementary strand as a template. Okay, using the complementary strand as a template. And then finally the last gap will be filled by the ligase. But the main question is that how does the damage base can be removed out by the glycosylase? Okay, the damage base, how it can be removed by the enzyme glycosylase? The damage base is in the inside, so how it is removed? They are involving in glycosidic bond formation, right? So how it is removed? It is accomplished by this flipping of the damage base. Suppose there is nucleotides, and suppose this is your damaged nucleotide. Okay, this is your damaged nucleotide. Only in one strand it occurs. So DNA glycosylase, what it does, it binds, and what it does is it flips that damage base, it flips that damage base to the outside. It flips that damage base to the outside. Okay, so if this is your DNA glycosylase, it flipped that damage base to the out of the double helix, and then it is followed by a cleaving of this glycosidic bond, but the base will remain intact. The backbone will remain intact. The backbone will not be damaged. It will further be removed by the your epi-endonuclease. Okay, so in this way your DNA glycosylase act. DNA glycosylase flips that damage base out of the double helix, followed by a cleavage of the cleavage means cut of the glycosidic bond. Okay, flipping the damage base, the damage base is rotated outside. Then the damage base is now rotated outside. I will show you the main picture now. The main picture will be more useful for you to understand. Okay, so this is your base excision repair pathway. So this is your DNA that you can see here. Okay, and this is your uracil. The glycosylase will cleave here, and the uracil has been damaged. Okay, and glycosylase enzyme cleaves here and makes them, see, glycosylase cleave here and makes them apurinic site. Sorry, uracil is a pyrimidine, so pyrimidinic site. Finally this epi site will be removed by the epi-endonuclease, and epi-endonuclease is actually it is cleaving inside to remove that rest of the nucleotide which was been damaged. And finally the gap will be filled by the DNA polymerase then. Okay, pol 1, it has been says, it says pol 1 in case of bacteria. In case of prokaryotes is pol 1, and with the help of their proofreading activity, exonuclease activity, and by synthesis it synthesizes that area, that gap portion, by using the non damaged strand as a template to fill the gap. And finally the nick will be sealed by the ligase enzyme. So here is shown the base excision repair pathway, the uracil glycosylase reaction. See it what it says. Uracil glycosylase hydrolyzes the glycosidic bond to release the uracil from the DNA backbone and to leave the epi site, apurinic site, or here in this case pyrimidinic site, then this epiendonuclease then cuts the DNA backbone at the 5 prime position of the epi site, leaving a 3 prime OH. Exonuclease cut the 3 prime position of the epi site, leaving 5 prime phosphate. Then the resulting gap will be filled by your DNA polymerase 1. So this is the case of your the action of base excision repair mechanism. Okay, so I have not much time. I will try to do the next video on your this nucleotide excision repair pathway, this NER, and I will also go for the mismatch repair pathway as soon as possible, and also go through the other pathway, like your in case of double stranded breaks, which type of pathway are involved. These three type of pathway are involved. Okay, you have not to read so much. Based on the syllabus you have to read only, you have to read the NHEJ only, non-homologous end joining, which is also you are studying in immunology in case of VDJ recombination. Then comes your the translation synthesis, the last try to save, at least to save that damaged base. Okay, so this much we have to study. um, okay.